So, guten Abend, sehr geehrte Damen und Herren. Ich starte zunächst in Deutsch. Mein Name ist Ruven Porz. Ich freue mich, dass ich heute hier der Moderator sein kann und derjenige, der die 2 Minuten einführende Worte über Effy Vayena hält. Ich selbst arbeite im Inselspital als klinischer Ethiker und bin Professor für Medizinethik hier an der Uni Bern. Und ich glaube, Sie sind vertraut damit, ich sage es trotzdem noch einmal, dass erstens wir nach den ungefähr 50 Minuten Vortrag eine kleine Pause machen. Und zweitens wurde mir nahegelegt zu sagen, dass hier Collegium generale ist. Für diejenigen, die irgendwie ein Stockwerk weiter oben etwas über die EU oder so, glaube ich, hören wollen. Jetzt hätten Sie noch Zeit zu gehen. Wir sind aber auch froh, wenn Sie da bleiben. Now I switch to English, but later the questions, die Fragen können wir durchaus auch in Deutsch stellen, but your talk will be in English. So we are very happy to have here tonight Professor Dr. Effy Vayena. And now I read out, because I really want to be polite and be precise with the title and wording. She is Professor of Bioethics at ETHZ Zurich. And she completed her education, Effy, as a social historian with a PhD in medical history from the University of Minnesota. And then she did a habilitation on bioethics and health policy at the University of Zurich. A keen interest in health policy led her to work for the World Health Organization. How many years, Effy, did you work? 7. Well, it's amazing that you then afterwards made your way back to the academic world, to the university in Zurich. And upon her return to academia, Effy was awarded a Swiss National Science Foundation professorship and founded the Health Ethics and Policy Lab. And I understand that this lab moved to the ETH in 2017. Now, I also understand that you are currently an appointed faculty associate at the Berkman Klein Center for Internet and Society at Harvard University. You're very polite, you wouldn't correct me, but I'm right so far, right? And she has published over 100 peer-reviewed articles, several books, chapter, co-edited books, and so on. And I think it's also important here in Bern to note, because the Swiss Academy of Medical Sciences is here in Bern, that she is an elected member of the Swiss Academy of Medical Sciences, and that she chairs the Ethical, Legal and Societal Implication Advisory Group for the Swiss Personalized Health Network for the Swiss Academy. Is that right? It's a joint project. And you told me that you travel a lot, if I understand correctly. She's currently also the co-chair of the WHO Expert Advisory Group on Artificial Intelligence, Health Ethics and Governance. So that gives you an impression that she does a lot. And besides that, she's also a very nice person. We're very to have you here, Effy.
Well, it's always a gift to be introduced by a colleague and a friend, so he will say the good things. And so thank you very much, Ruben, for the very lovely introduction. And thank you very much for the invitation to be here. And thank you very much to Sarah for putting up with me of not responding in time to your emails, but arranging everything for this nevertheless. So it's a pleasure to be here, and actually a pleasure to talk about a topic that it is very complex and very much at the moment in the discussions in the lay public, in the policy space, and in the regulatory space, and of course in the scholarship and in the research. So what I want to do in the next almost hour is to talk to you about ethics and governance of this technology of genome editing. But as I will be talking to you about ethics and governance, I would like to invite you to be thinking along these 3 themes: how we move between hope and fear in those kinds of technologies, how progress, technological progress and scientific progress creates a lot of uncertainty, but also how uncertainty about what progress is generated by every step that we're making in science and technology, And also something that I call the intimidation of disruption. This technology, CRISPR-Cas9, but also other related genome editing technologies are called disruptive technologies. And in some ways, they bring a kind of rapid change that I think intimidates the way we articulate our ethical discourse, but also the way we design our governance around those technologies. So try to think along those themes as I'm walking you through some of what's happening around the ethics of genome editing and also about the governance. Now, it's a really interesting time to speak about this topic because next week it's going to be what people call an unwanted anniversary. This person here is Jennifer Doudna, is one of the co-inventors of the CRISPR-Cas9 and a very vocal voice in the scientific and ethical and regulatory space. So Jennifer published last week a short comment in Science and with the title, CRISPR's Unwanted Anniversary. We have the anniversary of the Chinese CRISPR babies next week. That's what she's referring to. And it's interesting to look at the language she's using. So she does say that this is a disruptive technology that can make or break. So they say that there are key moments in the history of every disruptive technology that can make or break its public perception and acceptance. And for CRISPR-based genome editing, such a moment occurred 1 year ago, as I told you, an unsettling push into an era that will test how society decides to use this revolutionary technology. So the bold is mine. She has written it without putting emphasis. But I wanted to emphasize a few of the words she's used because I think it's important to see her calling this technology disruptive and revolutionary, but also of her to speaking about the importance of public perception and acceptance. And finally, thinking that what happened is an unsettling push, something that makes us uncomfortable, and so we have to deal with it. So Jennifer Doudna, and I'll speak a little bit about her because she's one of those scientists that has very actively engaged with the consequences of technology and with the ethics discourse. She was one of the first people to speak about, hey, we got this technology, what are we going to do with it? After her own publication, of course, but after rumors went around already in 2015 that Chinese scientists, once again, were genetically modifying human embryos. So there are a couple of publications that, and lots of rumors that, okay, we have the possibility to use CRISPR-Cas9, that there have been other methods in the scientific toolbox, but this is one that was cheaper, more effective, and easier to use. And the worry initially very quickly became, okay, what if we use this to modify our genome, not just use it for other applications? I'll make the distinction a little bit along the way. So after these rumors came out, and Jennifer Doudna and a lot of other scientists in particular started becoming quite vocal about what are we going to do, she was interviewed by a journalist who writes for The New Yorker. And it's interesting, she has also, later she published a book, but this extract at the time, it was the first time she spoke about this, it's a bit longish, but she talked, and feel free to read it, but the short version of this is that she spoke about a dream she has. Again, I think it's interesting to see how she pictures the whole story. A dream, she says, that in which somebody came to her and wanted a very powerful person that I wanted her to explain what this technology, how this technology functioned. So I said, sure, who is it? It was Adolf Hitler. That is Jennifer Doudna speaking about her dream, right? I was really horrified, but I went into a room and there was Hitler. So he had a big face and I could only see him from behind and he was taking notes and he said, I want to understand the uses and implications of this amazing technology. And I woke up in a cold sweat. I mean, I suppose anybody who dreams of Adolf Hitler has the same reaction, but she was involved because here's a technology that she helped develop, or she co-invented that technology. And it was quite a sort of a strong way to put this out. It was picked up and discussed in various settings. I think it was, again, a way of her to say that this is scary if it gets in the wrong hands, number one. Number two, we've seen things happening with people having wrong things handed in the wrong people getting access to technology. And of course, that I think resonated with some of the public sentiment about fears of technology. So she and a few other scientists, and you can see the names, maybe you cannot see the names, but very stellar list of people, went and out and put a paper, a call essentially, for what they named a prudent path forward for genomic engineering and germline gene modification, where they argued that, well, it's great to have this technology, but we got to be very, very careful. And we cannot use this technology to edit our genome. We can maybe use it or try to experiment with it for somatic changes, but we should not use it to change our inheritance and modify embryos, etc. This is one of the first pieces that came out, and made reference to something that we knew already from the conversation about recombinant DNA and the Asilomar Conference of 1975, which I'm putting here in order to show you that it is not the very first time that scientists see the potential of a technology that can go the wrong way, and they're getting together debating how they can actually prevent that from happening. In the Asilomar Conference, the conversation was about recombinant DNA. It was a couple of people who were involved later in the CRISPR discussion. They convened 140 scientists. They developed voluntary guidelines. The idea was then self-regulation. So we, the scientists, get together and decide how far we should go and then implement this regulation on ourselves. There were no ethicists and lawyers and public, etc. So this kind of approach has been criticized, and we know what happened. We didn't have a major release-related incidents, but we did have debates about, for example, GMOs, etc. Another example of interest, I think, again, of technologies that we're worried about, and this is my theme of oscillating between hope and fear. I'll come to hope. I'm starting with fear. Is in vitro fertilization. I'm old enough to remember the term of the use test tube babies, not IVF babies, which I thought it was very weird because I was a kid and I couldn't understand how a baby can be in a tube. But it was how we used to call these babies because it was just such an exotic thing to be happening. So when the first baby was born, everybody, first of all, to get to the first baby, it was very difficult due also to the discourse and objections. But then people were very worried about where we're going with this technology, how it's going to change and affect our morality and our lives. A very famous person was interviewed by the New York Times in 1974, before the first baby was born. And he said, if we go ahead with IVF, in vitro fertilization, right? We have a few million babies born today with in vitro fertilization, as all of you know. He said, if we go ahead, all hell will break loose politically and morally all over the world. And you might think, well, who is that person? Is that a conservative person or scientist who thinks, or not a scientist maybe, who thinks that way? It turns out that person is Jim Watson, who, as you know, was one of the people who got the Nobel Prize for the double helix, the DNA double helix discovery. So this, again, from the scientific discourse, we had at that time the conversation about how a technology like this, in vitro fertilization, is going to be something very scary and provocative. Now, thinking about, in coming back to CRISPR, what happened after these publications and the rumors about the germline, we had a number of applications that could be possible. And of course, you could see the scientific literature. If you look at the PubMed, the repository where scientists put medically related articles and scientific articles, you would see a spike because so much was going on. And basically we could imagine CRISPR being everywhere, plants, animals, humans. So much that the NSA, that in the United States, the Director of National Intelligence actually listed CRISPR as a weapon of mass destruction, along with North Korean nukes, which is quite a place to gain in that kind of list. But again, there was fear and there was hope. At the same time, with all of that and all the excitement, there were the positive news, which, of course, were about, am I sitting in your view, maybe? The positive news were that, well, we can use CRISPR technology for somatic purposes, forget the germline editing. We're not going to have embryos. We're not going to mess around with embryos, but we can have other ways in using the technology to actually treat diseases that we don't have another way to treat. And news came that there are a number of possibilities, and patients actually, groups of patients were very keen to find ways to benefit from the technology. I have a couple of quotes from patients who also participated in some of the meetings that the debate was taking place. Should we go ahead with CRISPR? How should we regulate it? Who can do it? Can anybody do it? Where do we draw the line? And people would say things like this. For example, Matt Woolsey, who was in one of the first meetings that brought together scientists and ethicists and regulators to decide what to do with CRISPR. So he does say, as a parent with an incredibly sick child, what are we supposed to do? Sit by on the sidelines while my child dies? There is zero chance of that. And he went on to say that CRISPR is a bullet train that has left the station. There is no stopping it. So how can we harness it for good? Now that's the other voice, the people who think that if there's anything that we can do, is that CRISPR? Okay, fine. Then we're going to do it because we are in pain, we have our sick children to treat. And of course, other people have commented on this, and these are just some of the voices. I bring them out here because very often we don't bring the voices of patients, but just our more theoretical academic perspectives. That's a patient with Huntington's disease. Again, we don't have it's a lethal disease. There is nothing that can be done at this point. So if there was a room, he says, somewhere where somebody said, look, you can go in there and have your DNA changed, I would be there breaking the door down. And these were very powerful voices in the discourse when scientists, regulators, ethicists were trying to say, well, no, you cannot go on with this research, or yes, you can go on. In the meantime, there have been a number of clinical trials that are attempting to actually test CRISPR technology, not in modifying embryos, but for somatic uses, which means ways in which they can treat diseases. There is a way, the clinical trials are registered in big registries, like the one, this is the American one, the clinicaltrial.gov, and if you look at this today, you find 30 registered clinical trials using CRISPR technology. Some have been suspended, have withdrawn, but it just shows you the activity at the moment, and some are currently recruiting. And only yesterday, because giving a talk for CRISPR, you got to watch the literature until the very last minute before you speak, because just yesterday, this is the 19th of November, there was news that the first CRISPR treatment for 2 blood diseases showed early benefits. We had 2 patients with early benefits that cleared from the disease in using CRISPR-Cas9 technology. And that was big news for scientists, big news for the companies that are backing this, and I think big news for the conversation. So that's the hope, and we're still already seeing activity, but we're also seeing early results, very small, but results that I think give people the interest and the energy to continue. I'll show you later why these kinds of applications might be a little less controversial. So I thought, okay, let's start with the less controversial ones. Because what has captured, I think, public imagination and the regulatory imagination is not so much the somatic approaches, but rather the germline editing. Shortly after the first conversation, the first days of the announcements about maybe the Chinese scientists fiddling around with embryos, very quickly afterwards there was a colleague in the US who published a book, and the title was GMO Humans. Now that's quite a provocative title, because we know about the discourse around genetically modified organisms, and he was trying to make that link that here we go from, you know, genetically modified potatoes, we're going to genetically modified humans. So a provocative title and provocative in order to initiate, I think, what he wished to be a provocative and challenging conversation. So I'll spend a little bit of time in telling you about what are the main concerns when we move from even the somatic applications, which is not all on the clear, but when we discuss germline editing. Now, it's interesting that we call this editing. And I think often the metaphors tell you a bit about what's in the background. So we talk about editing, and this is from a piece that was in Nature, actually, some interviews. It's like going into a Word document and basically replacing one letter. Maybe that instead of wind, you want to say wine. That looks very simple and something we do all the time. And also something that allows you to improve something. Like I edit my papers because I want them to be better. I don't edit them, well, I may make them worse at the end, but in theory, I edit them to make them nicer. So by editing, you introduce the idea that you make something better than what it is, and that's an evaluative judgment. But also that there is something that needs to be edited. So if something is fine, you're not touching it. You edit things that you think they're calling for some improvement. And the context, and I think the metaphor here has been an interesting framing of this conversation. Here we are, we're editors, so we can do that, something that needs improvement, and we can do it. So keep that metaphor in mind. At the same time, what we know in our kind of discourse and regulation is that actually we are not allowed to fiddle with that kind of improvement. This is from the Convention on Human Rights and Biomedicine in the late 90s, signed, that clearly states that an intervention seeking to modify the human genome may only be undertaken for preventive, diagnostic, or therapeutic purposes, and only if its aims is not to introduce any modification in the genome of any descendants. That's clear. So you can do something to prevent a disease and therapy, but a lot of countries have signed into this convention that doesn't let you go anywhere further. So essentially, we could just close the conversation there and say, well, we debated that in 1997, we agreed, and we should just stop. But it's hard to do that. And it's hard to do that because I think we've been kind of playing with this idea for a long time. In theory, not in practice, we had these conversations with gene editing possibilities, even with in vitro fertilization, with pre-implantation genetic diagnosis. Should we do designer babies? Should we not do designer babies? How far we can go? But we never came as close, technically speaking, at least in theory, not in practice, as maybe this. And there is in our culture so much of that. Those of you who've read Kazuo Ishiguro's book, Never Let Me Go. He got the Nobel Prize a couple of years ago. I think a very worthy Nobel Prize in this case. He has this beautiful story about precisely making people edit them, design them the way we want for a particular purpose. And we all, reading these books and seeing also movies with those kinds of themes, we feel that that is really not where we want to go. So there's a general sentiment that if we are going to fiddle with our generations to come, there's something wrong with it somehow. The difficulty is in articulating what exactly is wrong with it, and if there is a way to do things like that correctly. Now, despite our general sentiment of don't fiddle with the genome, we shouldn't make this kind of changes, we cannot play God, and all of the, lots of wording that's been used, a year ago, next week, a year ago, we had the bombshell in China. So a lot of people were gathering in Hong Kong for a CRISPR summit. And in that summit, they were supposed to speak about science and technology and about regulation. How are we going to regulate this technology? How are we going to go and draw the lines? How much somatic applications? How are we going to deal with the challenge? And sit there, and here, the Chinese scientist stands up and says, like, while you all guys are discussing how to regulate this technology and what's right and what's wrong, I just did it. I produced 2 babies which are CRISPRed. So I was able, he claimed, to modify the genomes of twins, and they're now born even. So the babies were born. This was not an announcement of a research project. This was not an announcement of something that's coming. That was something that had already happened. And he seemed to be incredibly proud of it. He said the project was, he called it initially an immunization research, because the babies that were going to be born, they were going to be immune to HIV. And because his claim was in China, HIV is a very stigmatized disease. We know in many other countries, HIV is now a chronic disease rather than a disease, a death penalty disease. But in China, it's a stigma. It's really important for people not to be able to acquire HIV, as if that was the one and only way through which people don't acquire HIV. But anyway, the point is that he defended his work, and he was put on the spotlight immediately. Imagine this. I don't know how many of you followed this. For some of us, this was like, where were you when this thing was announced? Because it's just such a crucial moment in the history of gene editing, but also in the history of bioethics and health policy. So what he said he did, at least he claimed he did it, and I'll say why I say he claimed, is that he made a change in this particular gene, CCR5, precisely in order to make the 2 girls, eventually they were girls, immune to HIV. At the time, because everybody got crazy about what's happening, I'll explain to you what this is. He also put up a couple of videos where he explained his intention. It was a really interesting. There are no videos to be seen at the moment, and I didn't download copies. But he's just saying how he's really trying to save those people, how beneficial this is, how correct it is, how patients really, really, really wanted that. And so just presenting a picture that was amazing in terms of, how he is a hero and somebody that is going to save the world, and a first, because nobody had done this so far. So he is first. This is unpublished data, but what we did with my colleagues from EPFL and somebody from ETHZ, we looked at Twitter traffic around CRISPR, the term CRISPR, from 2015 to almost 2019. And what you can see is in E, if you can see that E on the second, on the bottom field, E is the maximum traffic on Twitter in terms of mentions of CRISPR, and that's exactly when this event took place. We had CRISPR going on from earlier on. You could see there's some, some conversation here and there, and then bang, that spike. It's the highest, and it's the most concentrated activity. Everybody was talking about what He Zhankui did. Everybody was also speculating. He just didn't only change he didn't only try to change those embryos in order to be HIV immune. He maybe inadvertently enhanced their brains. So a lot of speculation and rumors about, well, maybe that's a Chinese project, maybe that is some real science fiction thing that is going on. Well, the thing is, we don't know what he did exactly because he didn't publish the paper. So the way we know whatever we know is when he was presenting and he was showing his slides in Hong Kong, people were taking pictures of his slides and then studying them in the labs to see whether that's the case, whether it's true what he's saying. But again, you couldn't say exactly if he's true. And there was even a debate whether should we publish such a paper. Now, this paper breaks international conventions. It's highly problematic. Should somebody, a journal like Science and Nature or whatever, any other journal, publish this? Well, today, the paper hasn't been published. I think he had a lot of other problems than because he was removed from his university, he got sanctions, et cetera, so China went after him, so he wasn't let on the loose. But what happened is that the issue is that we were not, even if he had submitted this paper, our scientific community wasn't sure how we're going to treat it. On the one hand, we want to know what happened because we speculate. On the other hand, do we endorse by publishing something that was universally considered to be reckless and unethical, because there is no safety standard to begin with for using such a technology. And very quickly, China had to respond. Of course, the conversation in the West was, well, China is wild. They're doing whatever they want. We are good. We're not doing those things. But China produced a lot of comments on the topic, the Chinese government at least, or at least in terms of a response in the public sphere. And they clearly suggested that they tighten their rules in order to make sure that whatever happens in the future takes a different, it's not done this way. Now, you'd think, okay, so we've got China probably trying to get under control. He has his 2 babies and 2 other pregnancies on the way. At least that's what he claimed. We're not sure what to do with it. So we keep thinking, and we keep writing papers about regulation, and we keep having meetings to think, okay, what should we do, and how are we going to do it? Meanwhile, we have another guy, Rebrikov in Russia. He says he plans to edit babies, but he has a better solution. Because while he wanted to reduce the risk of passing HIV, from his worry was that he makes it immune, so the risk is not passed on by the father. He says, okay, the risk is higher to be passed on the infection by the mother, so I'm going to make the change so that this is not possible. But he also says, I'm aware of the international debate, I'm not a rogue scientist, and I won't transfer the edited embryos without the permission of the regulator. And the regulator here is coming into the picture, because in this case, it is President Putin that he has to decide. Now, this is a really interesting development, because while in China we don't know who's going to make the decision, we know who is going to make the decision in Russia. And it turns out that Putin has set up a whole group to explore the ethics and science of this technology and make a decision. Now, the group is headed by his daughter, who is a doctor, by the way, and apparently very interested in the ethics of science and technology. So it's really interesting to see how this discourse is moving between countries. And we don't have a verdict yet from Putin's daughter, but I think we're reassured that he is involved. So let me just go through some of the issues the ethical issues that have been raised while all of this, partly theater and partly reality, is going on. And this nice summary has been provided by my colleague who recently wrote extensively on this, Dr. Blasime. So I'm using his slide here. So the first set of issues that have been raised is about safety. Off-target mutations, for example, trying to do one thing. By doing one thing, you make inadvertent changes that you didn't plan, and therefore the safety of these changes has not been tested. You don't know what we are doing. Unknown effects of the targeted mutation. Let's say you know what you're doing and you're making a change or that editing, but you thought you knew enough about it, you just didn't know it all. or you can have containment in the lab, so very practical kind of sorts of safety issues. And if safety is not clear, then we do have we cause harm. And that violates immediately the do no harm principle. So we have already not, at the moment, we have not resolved the safety question. And that's a very, very preliminary and basic question to address. In germline, in somatic, we have that, again, this can apply both in somatic therapy. For example, if you have mutations associated with polygenic diseases that maybe have protective effects of the thing you're exactly trying to change. But in germline, the questions are even harder to tackle. For example, human dignity, the integrity of the human genome, which some consider to be the basis of human dignity. And the Oviedo Convention that I mentioned to you earlier is actually structured around that. The idea that we might have completely unpredictable consequences on future generations. That's very much linked to the safety question. If we don't know what we're doing, we might end up landing in a very bad place. Concerns about how we all trust science if things go wrong. The whole idea of designer babies, that's not a new conversation, but here became closer to reality. The concepts of genetic narcissism, or commodification of children. We make people to our specifications. The perennial problem of whether that kind of research would require destruction of embryos, which gets us into conversations that we have had for a long time. These are embryos, actually, I thought that was quite interesting. I don't know how we store embryos here, but I think that was quite an interesting way of storing embryos. That's from a facility in Moscow. And of course, the question of eugenics, very linked to the idea of designer babies and making people to our specifications. This girl here was again quoted in a paper in Nature about looking at people with disabilities who live with them and lead certain kinds of lives. So she has a hereditary blindness. Apparently she was asked whether she would wish to be corrected, to have a gene correction, so her own children are not blind. And her life, which seems to be fine for her, was a very, very clear no, because she has a life that she doesn't think is not worthy of having the way she does. And so it's interesting when you enter the conversations about how and what you design, or where disease ends and enhancement begins, that we have often, except with very, very clear cases, difficulty in determining. So the comeback of eugenics, inequality of access, another big question. For example, who would be able to go through the process in order to get what they want? Would this be something that's available to everybody? Let's say we cleared the safety. Let's say we determined that it's okay to do X or Y. Is this going to be available to everybody? Or just some people will have access to making themselves better or their children better, but not everybody will have that. There was a conversation also about informed consent, that people had not consented to be CRISPRed, which it always puzzled me because I'm not sure how we get consent from people to be born. But certainly in this case, it was part of a few, discussed in a few papers. Discrimination and stigma for those people who are right now living with the condition, like the girl I showed you earlier. If we can fix that, is it okay to be alive today having that problem? And of course, the general pressure to conform. If we can fix that and we decide it's a good thing to do, does everybody have to conform with that, whether they can or not, or that they want or not? So these were the arguments against against the ideas of genome editing in particular. But there have been a number of conversations about what is actually going on for CRISPR. It is not just a horrible thing that we can destroy our species, it's going to destroy our species, but rather perhaps something that we might even have, it might improve us in ways that makes us also morally better, not simply physically better. And so the response to the question of safety was, wait, there's so much technology that was never safe, and we tested it slowly until we thought it is safe. So something is not safe doesn't mean it's out of the question, because we just don't know where the safety boundaries are going to be. And we have some ways of doing dangerous experiments with humans that actually have been quite then, as a result, positive to have. Why should we now practice some form of exceptionalism from CRISPR? What's the reason? Is the threshold of safety here different? The argument about human dignity would have to lie on the fact that our genome is stable. And therefore, we have to keep it that way, which again, it's hard to argue that it is. It's just that we didn't necessarily intervene, although I don't think, it's correct to say that we haven't intervened so far in many ways to our genome. We just didn't do it in the lab, but we did it in different ways. And when it came to somatic and germline therapy, again, if it's to save lives, then if we think that we can save lives and improve people's lives, don't we have a moral imperative to do that? Why are we doing this with all other technologies and medical therapies? Why then leave this out? In fact, there are a couple of colleagues of mine, Julian Savulescu in particular, who's been arguing for not just making these technologies permissible, because they can be not debate the permissibility, but rather debate the fact that they might be moral imperatives. And again, in terms of reproductive choices, people argue, well, natural reproduction has a lot of risks. It's not risk-free. People who argued, we don't need CRISPR because we have in vitro fertilization and pre-implantation genetic diagnosis, so we already have enough tools that don't make us cross safety lines that we don't know how to deal with. Again, those technologies are not risk-free, although we're just more used to them than we are to others. And again, another concept that has been in the literature for a while, that of procreative beneficence. The germline editing aims at giving people the best possible chances. Aren't we doing this anyway, not in the lab, but in all sorts of ways? We try to have, our children to put them in good schools, to give them good books, to give them good food. We want to be better, to be smarter. We try that. We just don't do it in the lab, but we do it in so many other ways. So why is this moral difference if we do the same thing, we're just using different means? And again, it could, with the consanguineous marriages, this would have been, in certain places of the world, quite a solution. So we don't have a verdict. These are arguments for and against, and they have been entertained for a long time, and particularly in this debate about CRISPR, among very intelligent people, and very eloquently. But we do not have at the moment a conclusion as to which is the strongest argument. They are very reasonable, and I think they have to be weighted, but it is unclear whether we have a strong point in saying either no or yes. And that's always the case. It will depend, and we have to maneuver in the middle space. Now, the question is, if we are in that kind of conversation, how are we going to govern this? What are we going to do? Because we're not going to just sit around and look. Some people are in labs doing things. Patients are thinking they're having something to get and are quite vocal. Regulators are trying to decide which way to go. And the public, again, of course, reads in the news, CRISPR today treated maybe 2 people with 2 blood diseases, so that's a good thing. And then some Chinese rogue scientist produces babies that they might be immune to HIV, which we don't know, but they might live a much shorter life because of the changes he made. So how we govern? And by govern, I want to explain what I mean, what I mean is that this system this distributed web of actions and processes that we as a society have a way to exert control over a given domain. That domain is biomedical science. And again, I want to make sure that there is a distinction between governance and government, because in governance we have a multiplicity of stakeholders, and we have a lot of different practices. And again, because we can, with governance, at least in my understanding, reach beyond political institutions and legal instruments to include more informal tools like self-regulation, like guidelines, like codes of conduct. So if we have this toolbox, how are we going to use it? And here are some examples of how we have been trying to use it, which is basically making policy. So a couple of organizations have tried to come up with some suggestion. Again, these are not law. These are recommendations, and this is soft policy. The US National Academy of Science and the National Academy of Medicine, they set up the Human Genome Initiatives, and the National Council of Bioethics. I'm just taking these two. There have been a few others, which is a very well-known, thoughtful group of people in the UK opining on issues and challenges in bioethics. And I think if I were to kind of distill what's going on in those kinds of conversations, where we stand, I would say that the consensus is right now, probably proceed with caution, and for some people that is translated as a moratorium, and for others, no. And there's a debate about the word, so I'll get to that. So take a look at what these two bodies have tried to say. It's a lot of text, you don't have to read it, but I tried to distill it in terms of what is the important things they're trying to put forward. This is the Americans, the NAS and NAM. It's the Valentine's Day report because it was reported on the 14th of February in 2017. That's the only, I think, relationship to Valentine. But what it says, this text and these recommendations, basically is these points here. Safety is paramount. We really need to establish safety, otherwise we cannot do anything. And we will establish safety by trying it carefully. The thing we agree upon is if there is a therapeutic purpose, then we can contemplate it, but not if there's no therapeutic purpose. So we seem to, and I will show you some other data, when it comes to therapy, treat a disease, somehow we seem to have more consensus. Oversight. We need somebody to looking over those people who are doing that stuff. And that is a message across the board. And we need to be transparent. Unlike He, who said, okay, hello everybody, I did it, and nobody knew about it. It turns out some people knew it, but it was not publicly known, it was not vetted, it had no proper oversight. What we need to do is have to have transparency about these activities. And one thing that I'll come back to, public input and dialogue. All these committees recommend that we have public dialogue about those matters. This is the Council of Bioethics. Again, a long list of recommendations, slightly differently worded. But here, what we get out of NAFLD is safety again, paramount. We need to establish safety. It makes sense. That's how we proceed in medicine. But the NAFLD talks about a few other things. Social justice. We can't proceed with a technology that we cannot ensure that it will be accessible to all. Solidarity should be a principle guiding these developments. Oversight, again, we need somebody to look over the shoulder of those who practice it. The UK has an interesting mechanism, the Human Fertilization and Embryology Authority. It does a case-by-case approach. So the UK has a model that can actually be used in similar situations. The rest of the world has very different models, but it's I think the UK's one is very unique. And again, the NAFLD says public dialogue. Let's talk with the people about what to do. That's what you will see everywhere in the literature. So I'm going to spend a little bit of time on that societal dialogue because I think it's important. Now, societal dialogue, I want to say, it's very different from studying the society. And what I will show you is that we study the society. So these are just some studies which asked people, what do you think? Should we use CRISPR and gene editing for somatic therapy? Should we use it for germline therapy? We should do it for enhancement. or not, clinical use or whatever. Give you those lists. So we scientists study you, the people, and try to understand what you think. That's kind of different from dialogue. That is a study. But nevertheless, we try to get a sense of what's going on. And what we see in most studies is that actually people, whoever is in those studies, they are okay with somatic therapy. They are okay with germline therapy, at least most, right? This is not accurate. This cannot tell you. It gives you just in broad strokes a picture. People are a little more concerned about enhancement, whatever they understand as enhancement, because we can open up the conversation. What is it really, enhancement? And then again, the degree of disease and seriousness also changes the views. I would say from the studies we have seen out there, therapeutic purposes, fairly well accepted at this point. Enhancement and people understanding design, choose the blonde baby versus the dark baby or the blue eyes to the brown eyes. I think people in general do not think that is a wise application. And there are various studies that have looked at that. And again, I don't think, we don't have a meta-analysis, we don't have another systematic way, but I would say that's more or less in the general attitude. And again, interestingly enough, we have to study different groups. There are the patients, there are people with particular diseases, there are healthcare providers, there's lots of different groups. But this again, I want to say, it is not dialogue. This is some people studying other people and trying to understand their views, which is important. Interestingly enough, if you look specifically, these are some qualitative studies that have been done, not just asking people in a survey, but trying to get a better sense of what they really think. A lot of patients thinking about concerns and societal implications, they do raise things that I think have been included in the conversation in the policy analysis. For example, whether we become less tolerant, whether we have less diversity, what happens about the idea of having a perfect model of a child, things again that are not brand new. What we pick up in surveys and in the qualitative studies are it's a discourse, it's issues that we have been discussing, I think, for a long time. I'm going to show you some attempt to public dialogue, which I thought it was quite interesting. It's 2 months ago, and this is from Twitter. The National Academies of Science put out that tweet. They wanted actually to ask people to take a quiz, which means awareness, like, okay, here, let's talk about it. What do you think of that and the other? And this is how they introduced it. Dream of being stronger or smarter? Do you dream of having a top student or a star athlete or a child free of inheritable diseases? Can human genetic eventually make this and more possible? Now, if I read this, I'm thinking, wait, do I dream of being stronger? Sure. I go and spend time in the gym trying to be stronger or eat better. Do I want to be smarter? Definitely. Do you dream of having a top student? I'd love for my kids to be top students and top athletes. Who in this room doesn't want that? We all want that. We're trying that. And we're doing it with other means. And then, or do you want a kid free of inheritable diseases? Yes, we do. We've been doing this. We have been trying to have our kids free of as many inheritable diseases as we tried. And in some places, I come from a place in the world where we tried to get rid of beta thalassemia. My parents had to get tested and see if they're both carriers of beta thalassemia, and they had decided that if they're both carriers, they're not going to have children. We cleaned up that particular mutation in that part of the world, so now we don't have kids with that. Did we mess up some other ways? Maybe. I don't know. We don't know. But we certainly did not want to have that disease. We did not want to have that inheritable disease. So what happened with this is that tweet sounds kind of funny, reasonable, has also the emojis, which make it a little bit more jazzy, but they got a lot of heat because they said, do you really want to tell people that gene editing is going to make you stronger and smarter? That is wrong. So they had to pull the tweet down. It was a bad day for their communications department, that is the National Academy of Science, because a lot of people felt that this is almost insulting. Telling people with CRISPR you can do all those things, that is the wrong footing for a public conversation. This tells me one thing, which is that we don't really know how to have this public consultation and this public discussion. They try this, it's really wrong, say the academics. where else has been public discussion going on? Because I haven't seen any. Some journalists write something, some people write a letter to the editor, and we're there. In the meantime, there have been some other forces put in place. This is the World Health Organization, who said, okay, we're going to solve your problems by establishing a committee that is going to tell us what to do. And they did that again last year, a year ago, in December 2018. It's a global committee that is going to issue global guidelines. This is not law, this is just global guidelines, soft law approach in how to go about CRISPR. And what they've been doing, that's a very recent thing. What they did last August, they decided the way to go about this is actually to launch a global registry on human genome editing. So their idea was, we are not going to exactly say what you're going to do it or not yet. We're going to put a number of conditions, but let's be transparent and let's watch the space. So we have a global registry where anybody who wants to CRISPR anything will just, in the human genome, then it has to be registered here. And if it's not registered on that list, it cannot be published, it can be considered a violation of an international code, et cetera, et cetera. And only again, a couple of days ago, the two chairs of the committee, who is Hamburg, who is an ex-FDA commissioner, and Cameron, who is a South African justice, they put out a statement about where they are in the process. And it doesn't look that very far. They're only as far as to say they were going to establish the registry, but they said this is how they're going to do the rest of their work. And you can see this is the most impressive list of ethical principles and ethical wording. There is nothing left out. Transparency, inclusivity, fairness, responsible scientific stewardship, social justice, and the contribution will be a result of global and diverse viewpoints, good practice, and scientific legal conduct will maximize benefits, minimize harm, and allow everyone equal access to opportunities and potential benefits. This is a dream. I hope they make it. But it is such a dream. I mean, it would be the first time we got all these principles in one place and in one policy, and it worked. But it shows you also the struggle. I think we got the principles, we got the will, but we have difficulty in sorting this out in a way that it's going to be actionable. And we have to remember that we are living in new geopolitics. This is here from the Global Innovation Index that the World Intellectual Property Organization published a couple of months ago. In the red dots, you see where the scientific activity is these days, but you also see where the noise is. Lots of stuff is going on, but here are the clusters with the actual action. And you see the clusters are not any longer just where we knew them to be, which was in Europe and the United States. There are a lot of other people that are producing science. And I think it was very interesting to see again that China, these numbers are from China. In 1997, there were 2% of all patent applications came from China. In 2017, 44% came from China. That tells you a little bit about who is innovating and how. And it's interesting. This is not a bad thing. It's just that the world looks different. And I think when we try to do policy and figure out which government allows what to happen and where, we need to be aware of these new dynamics. So I think I've run out of time, but I will just use, have I run out of time? Kind of, a little bit. Okay, almost. All right, so I'm just going to have just my conclusion and what I think is I want to kind of the take-home message from this lengthy narrative here. In terms of governing, I think we are moving slowly. As you can see, even the national organizations, international organizations, they put out documents and wishful thinking, but we haven't seen something which is very, very actionable. And I think part of this is actually because that's the process. The process is such that it cannot necessarily go faster. It certainly cannot go as fast as science is going. The second point is that I think we have a little societal dialogue. Every single body that has talked about CRISPR, the scientists, the regulators, the ethicists, everybody talks about societal input and societal discourse. And I think we're lacking models for that deliberation. We don't have them. We study people, we ask them questions, then we don't know what we say, well, they're often not representative samples, etc. But we need different models if we want to have serious societal dialogue, where we also listen, but we also tell people. I think it's interesting how we continue framing the conversation as to, and I'll give you two examples. One is the metaphor, edit, for example. The other is the Chinese rock scientists versus the very nice people in the West. That is also not a useful framing of the conversation. We have our fair share of doing funny research. So it's interesting to see how we put that conversation going on. The geopolitics and the scientific arms race that we experience right now, China wants to be first, the US wants to be first, Europe wants to be first, everybody wants to be first, cultivating a culture in science that actually is leading people to make shortcuts. And I think that's a highly problematic place. If Putin decides how gene editing is going to go ahead, we are elevating this to a political discourse and not a scientific discourse. And so I think that is something we need to keep in mind, and I find it quite problematic. And finally, I think it's something that we've been grappling with for thousands of years, and here is just another example of how we do that. We need, slowly or fast, to be defining what we mean by progress and what we want progress to mean for us. Is it progress the next fancy thing where we show how capable we are by changing genes and creating people who can live longer in the moon? Or is progress something that increases the quality of life and meaningful life for all people on this planet? And I think that's still, it seems to me, that's still a question that we are debating, and it depends, the answer to this question also will have an impact on how we answer the governance of the CRISPR question. And I'll stop here. Thank you.
Thank you. So, I understand we have a quick break, right? 1 Minute Pause.